Well, thanks so much, everybody. I'm Lisa Morici. I'm a professor in the School of Medicine here in the Department of Microbiology and Immunology. I'd like to thank the organizers very much for us for gathering everyone here today and for giving me the opportunity to talk to you about some of the work that we're doing at the School of Medicine, and in particular, our program in vaccine development. So many of you might be asking yourself, well, the title of my talk is one adjuvant, two responses. What do you mean by adjuvant? So it may surprise you to know that actually most vaccines require adjuvants for their ability to work. And so you can think of an adjuvant as sort of an immune enhancer and an immune stimulant. And so vaccines really require this component for their action. And so we have a program where we're developing new adjuvants. And what's really important about the adjuvant is that it really determines the type of immune response that you get to that the vaccine delivers. And so you've probably heard a lot about antibodies and T cells because we're all well versed in the COVID vaccines now. And so really an ideal vaccine will engage both arms of the immune response, both antibodies and T cells. And of course, we want our vaccines to be safe, to be adaptable, and to be highly effective. And the adjuvants that are a part of that vaccine also can contribute to those. And so the current problem that we're trying to address is that basically current adjuvants that are used in vaccines today do not really promote all arms of the immune response. So we have adjuvants that are really good at promoting antibodies. We have adjuvants that are pretty good at promoting T cells, but we don't have adjuvants that can really help vaccines give us all of those immune responses. And so the solution that we're working on here at Tulane is called T-vant. And this is an adjuvant that we've developed based on outer membrane vesicles that are shed from bacteria. So it may surprise you to know that bacteria shed these vesicles, but in fact, all living cells secrete vesicles. So this is a natural phenomenon. And so what we do is we harvest these vesicles from the bacteria. They're basically very, very tiny, tiny nanoparticles, much like the lipid nanoparticles of the messenger RNA vaccines. And so they're really good, potent immune stimulating complexes. We can manufacture these. We can deliver these with any type of antigen. So a whole microbe, a piece of a microbe, a little tiny peptide from a protein. We simply mix it with our adjuvant, and we can deliver that as a formulation, and it's highly effective. Another important aspect of our adjuvant is that we can deliver it by any route. So most vaccines are given intramuscularly, but we can deliver this adjuvant orally, subcutaneously, intradermally, and importantly, intranasally, which I'll talk about in a little bit. And again, I just want to really emphasize the point that this adjuvant really drives all arms of the immune response. So this is just a table. I know it's a very busy table, but I just want to highlight for you alum or aluminum hydroxide at the top here. This is used in most of the licensed vaccines that we have against diseases like diphtheria, tetanus, pertussis, for example. It's a highly safe adjuvant. It's been used for more than 50 years. The problem with alum is it really only gives us great antibodies. And so what I have here across the top of the table here, so you can see we have CD4 T cells, CD8 T cells, and then CD4 T cells come in all types of flavors. So this is Th1, Th2, Th17. I'm not going to get down in the weeds about those, but basically what you can appreciate about these is that they all are good at doing different things. So some are good at helping fight bacteria, some are better at fighting viruses. And so you can see that while alum is really good at promoting antibodies, it lacks other capacities. And then we have some newer adjuvants that have come online in recent years. So ASO4, ASO1B, MF59, and CpG 1018. These you can see can check some boxes. So for example, we can get better antiviral responses with some of these adjuvants, but they don't check all the boxes. T-vant checks all the boxes. So we can get both antibodies, we can get T cells, and we can get every flavor of T cell. And what's really cool, and I know all of you guys appreciate the idea of intranasal vaccination now, because we're hearing a lot about it in the media now, that we can't stop this SARS-CoV-2 virus from circulating the globe because we can't block its transmission because it still colonizes the upper respiratory tract. So these messenger RNA vaccines are fantastic. They save lives. I'm a big fan, but they can't get rid of the virus here in the upper respiratory tract, particularly the nasal tract. So there's a real interest in developing intranasal vaccines. What's the problem there? We don't have any mucosal adjuvants. None of these adjuvants are approved for mucosal administration. And so if we need a mucosal adjuvant, we think T-vant can perhaps fill this need. And so I'm going to show you some data just briefly to show you what we're talking about here. So this is a comparison between an alum adjuvanted spike vaccine, so spike being the part of the SARS-CoV-2 virus that all of our vaccines target against COVID. And so this is a comparison of a alum adjuvanted spike vaccine that's given intramuscularly to a T-vant adjuvanted spike vaccine that's given intranasally. Now, this is in rodent models because we're not in clinical trials yet. But what I hope you can appreciate from these graphs is that the alum adjuvanted vaccines do not give us antiviral T cell responses in the form of interferon gamma secreting CD4s or interferon gamma secreting CD8s in the lung. So when you give something in the muscle, you don't really get anything in the lung. However, if we give T-vant, we get great T cells in the lung against that spike protein. If we look at antibodies, again, we know that alum can give great antibodies in the blood, but we're looking in the mucosa here. So this is IgA. This is a secretory antibody that's found in the mucosal tissues, so the gut and the lung. And you can see that alum, we don't get any IgA in the lung fluid. But if we give T-vant intranasally, we get great spike-specific antibodies in the mucosa. So this is a real advantage of our adjuvant just compared to alum in this example. In terms of our data package, we've done a number of safety studies. We've shown that the adjuvant is safe in a number of models, including waxworms, which is the smallest of living forms, if you can think of that, mice, non-human primates, and then this human in vitro mimic system. So this was a system developed by Vax Design. It was acquired by Sanofi Pasteur, and they put all of their vaccines through this system to sort of gauge whether they're safe and non-toxic. We now have GLP toxicity studies that are underway in collaboration with LabCorp, so we'll have formal GLP tox results soon. In terms of adjuvanticity and immunogenicity, we've shown that they're potent activators of dendritic cells, which are the cells that present antigen to the immune system. We show that we can get mucosal and systemic, so in the blood responses with this adjuvant, we get all arms of the immune response. Again, we've shown this in a number of models, rodents, non-human primates, and the human surrogate system. In terms of efficacy, we have protection data in models like pertussis, salmonella. We have some ongoing studies with the FDA right now for pertussis and non-human primates, and we've transferred the manufacturing to Catalent, who has shown that it's scalable, it's reproducible. In terms of cost, it costs about $200,000 to produce 1 gram, but that'll give us 1 million doses. So very affordable. And we have GMP manufacturing anticipated early next year. This is our timeline. We've done a number of preclinical studies. Again, right now we're focusing on GLP tox studies and early GMP manufacturing. And then, of course, we hope to move this eventually into phase 1 clinical trials. Just some highlights. We do own the IP here at Tulane. We're very interested in collaborations with pharmaceutical partners. Happy to collaborate with anybody else who has an investigational vaccine and any spin-out opportunities. And I'd like to highlight that this is a collaboration with my partner in crime in the back there, Dr. James McLaughlin. We'd be happy to talk with you more about the capabilities of T-vant and just talk vaccinology if you're interested. So thank you very much.
Any questions?
Yes.
Storage requirements, et cetera?
Yes, great question. So this particular adjuvant is stable at well, you can freeze it at -20, -80, but importantly, you can lyophilize it. And it's stable for probably at least a year at room temperature lyophilized, perhaps even longer. We just haven't done those studies. You're welcome.
Other questions? Comments?
You mentioned that it's derived from membrane vesicles from a genetically attenuated microbe. Is there anything else you can add to that?
In terms of source, or are you worried about LPS toxicity or? Yeah, so this is a genetically modified strain of Burkholderia, which is a soil organism. We can work with it under biosafety level 2. It is also a naturally attenuated lipid A species. And so we don't have to worry about toxicity in removing LPS from the vesicle because we get natural adjuvanticity from that without any endotoxicity. How did we come about developing vesicles? So this use of vesicles as an adjuvant, we believe is novel. There are vesicle-based vaccines out there. So Bexsero is an example. It's an FDA-approved vesicle-based vaccine against Neisseria meningitidis. And so we think, there's precedent for FDA licensure through that. There are other companies such as GSK and Intravac who have OMV-based vaccines. However, to my knowledge, to James's knowledge, I believe they're not using these as adjuvants. Our platform has a number of advantages over their platforms, which I'd be happy to talk with you about if you're interested. Thank you.